So this is my summative presentation, just a recap on everything that I have studied about this system for this semester. So it is the TsetseFly, and my hook is a tiny trio: the fly, the bacteria, and the balance of ecosystems. So here is a table of contents just to orient ourselves. First, we're going to talk about a system overview. We're going to talk about this system and also touch on African sleeping sickness. Then I will give a detailed introduction, going over the host and the symbionts, talking about discovery, transmission, and establishment within the two symbionts, which is Wigglesworthia and Sodalis. Then take a look at how Sodalis aids in vector competency. We will touch on dysbiosis when things go awry in this system. We'll talk about the immune system and immune response, talk about Wolbachia, which is a third symbiont within this system, and then last, give a summary on everything. So, who is in this system? First, we are going to talk about the host of this system, which is the tsetse-fly, and its species name is Glossina mortiscens. We will also talk about the two symbionts that we focused on this semester, which is Wigglesworthia and Sodalis. So they have two different functions within this system. So Wigglesworthia aids in metabolite synthesis, and Sodalis is going to aid in vector competence. These both symbionts, they benefit the fly, and then the fly also provides exchange in currency to both symbionts. We also recognize that the tsetse is a disease vector as it transmits Trypanosome brucei, the parasite that is responsible for African sleeping sickness. Why this system is important is because of its strict blood meal that the fly takes, and it does take it from vertebrates. So I thought this was important to study as it has a strict diet, it targets strict organisms, and in order to treat this disease that it transmits, it's important to understand who is doing what is helping this system, what areas of the life cycle can we target, and how can we aid in treatment. I do want to turn our attention to the schematic on the side just to get an idea of where these symbionts are located within the system. So if you take a look at that blue band in the center of the fly, that is called the bacterium, and that is where you will typically see Wigglesworthia reside. You will also see it within the uterine pouch and the milk glands further down the fly, as we will talk about that later. We also see Sodalis located around the fly, and it's called the hemolymph, so the lymphatic system of the fly. You will see Sodalis circulating in, and you will also see it localized to the uterine pouch as well as the milk glands for feeding its young. So let's get into the disease, African sleeping sickness. So we typically see this disease localized to sub-Saharan Africa. The parasite named Trypanosoma brucei, and it does infect humans and livestock. So there are two strains. There is a long, slender strain that is replicated asexually, and this slender strain is often easily detected by proteases within the tsetse-fly and then degraded. The next strain is short and stumpy, and this one does not replicate within the fly. However, this strain can evade the host defenses and live longer. So there are two subspecies under T. brucei, specifically noted as T. brucei gambiense and T. brucei rhodesiense. So gambiense is going to account for about 92% of the cases that we see in Africa, while rhodesiense is going to account for about 8%, and this is a more rare strain of trypanosoma. Some symptoms that we would see in those infected would be fever, headaches, fatigue, and swollen lymph nodes. If it does travel up to the brain, we can see severe neurological issues such as drowsiness, confusion, lack of coordination. And neurological symptoms are normally present in a late stage of treatment if it has gone undetected for a while. And it can also be fatal if not treated. Some treatment would include medications such as melarsoprol and fexinidazole. But the best thing we can do for this is prevention, which would include the incorporation of bed nets while sleeping, wearing thick clothing to protect our arms and legs, and also some insecticide to spray around the area. So first up, we are going to talk about the host of this system, the tsetse-fly, also known as Glossina mortiscens. It is a biting fly, and it is known to transmit African sleeping sickness, as we have already discussed, that it is the vector of Trypanosoma brucei. Some interesting fact about this host is that it does birth youngs that are larger than the full-size adults. Um, this is made possible because flies do not have a pelvic girdle that mammals do, and this pelvic girdle often constrains the size of our young. So we give birth to our young that is much smaller than our size, but because they lack that pelvic girdle, they are able to grow a young that can be their size or even larger. They are known to prioritize themselves as quality over quantity, as they produce one baby every 10 days. While they are able to do this, they produce a milk in their uterus, and they feed that baby inside of them for about 5 to 6 days before they give birth. This was very cool because I did not know that a fly would be able to produce milk, something similar that we mammals do. And this is what we call vertical transmission, as the mother gives birth to its young and it transmits its symbionts, its microbes, to its young through this milk production that it gives. So now that we have established our system and we have talked about the host of this system, we're going to talk about the first symbiont, Wigglesworthia glossonidia. So it is an obligate intracellular bacteria. It is localized to the bacterium of the tsetse. It is a gram-negative, rod-shaped, and it lacks a flagellum. It is located in the cytoplasm of bacteriocytes. It is in that U-shaped bacterium in the midgut. It metabolizes B vitamins for the fly. So when the fly takes in its blood meal, it is unable to get the nutrients from the blood meal on its own, and that's where Wigglesworthia comes in, as it synthesizes and breaks down that blood meal and provides B vitamins for the fly, amongst other metabolites. It is transmitted to the inner uterine larvae of the tsetse, and that is how its young is able to get it via vertical transmission. And these figures I got from Burgess' manual on the right hand side, the first panel is just to show that U-shaped bacterium where Wigglesworthia is, where it resides. The next panel over is just showing the bacterial sites and the cytoplasm. And the bottom one showed the actual Wigglesworthia bacteria, and it showed it when they treated it with a certain antibiotic, how the morphology was different and it kind of looked a little degraded and fleshed out versus a regular bacterium seen within the fly system. All right, so getting into system discovery, this relationship was established about 50 to 80 million years ago. In 1929, Sir Vincent Bryan Wigglesworth, he is the one who actually described this symbiotic relationship, and he saw it to be an ancient, obligate, mutualistic relationship. The bacterium was characterized in 1995, the genome was sequenced in 2002, and their original understanding was that it was a nutritional relationship. The Wigglesworthia was able to synthesize the metabolites for the fly, but then they evolved their thinking and they were able to realize that it's actually essential for the host immune development and ultimately host survival. So, getting into transmission, we recognize that Wigglesworthia is vertically transmitted from its adult host to the young. It does exhibit viviparous birth, which means that they grow the live within themselves and then they give birth once it is ready. Just kind of how, like humans, we grow our fetus in our bodies and then we give birth 9 months later. That's kind of the same thing that these flies exhibit. We also see vertical transmission through milk secretion, and it is to aid in larval nutrition. The Wigglesworthia is intracellular to the bacterium, and it does have this recognition protein called PGRPLB, which is seen in the milk glands, and this is what helps the host to recognize this symbiont as a friend, and that it evades the host immune defense response. So the figure on the right is trying to convey the abundance of the recognition protein at key times during larval development. It first looked at expression in the milk glands versus the larval glands, and we see a higher abundance in the larval band. Then they looked at the pupae at 24 hours in the early period of development, and then later on 3 weeks in the later period. And they see that as the larvae developed, the abundance in the expression of that protein is also increased. In the last two comparisons, they looked at the adult flies and noticed the same trend. the higher abundance at 7 days rather than 2 days. The overarching theme is just that as the fly continues to develop, it also increases its recognition protein to protect the symbiont. How did this relationship become established? We first have to recognize that both parts to the relationship is lacking something that the other can provide. So, taking a deeper look, Wigglesworthia is able to synthesize metabolites. In this specific system, it does metabolite B vitamins from the blood meal. We have seen that the genome has been severely reduced as it has evolved with this system for a very long time, but it did keep the enzymes that are able to encode for the synthesis pathway of B vitamins. So this is what Wigglesworthia provides to the host. Now, on the other hand, the tsetse provides protection from the host gut immune response. It does also give this symbiont a steady nutrient supply as its only meal is blood meals. And then it does have a multivitamin transporter that allows to pick up the metabolites and be able to take it across the cell membrane for the host to be able to use those things. This figure does show the different properties of the Wigglesworthia genome. The main reserved genes are the metabolism of cofactor and vitamins, and the pie chart shows the abundance of the metabolites that the microbe breaks down. And it does show that the B vitamins are the main group of metabolites, but there are some other things that it synthesizes. So this kind of confirms what we're saying, how Wigglesworthia is providing metabolite synthesis to this system. How does Wigglesworthia influence development in the system? First, it is essential for proper functionality. It is known to enhance the immune system maturation in the tsetse, and it does aid in fertility. When Wigglesworthia is gone, we do notice deficient cellular immune response in the adult fly. It is going to cause some disruption that's going to impact carbohydrate and amino acid metabolism. And we also recognize that the fly will eventually become sterile, even though they exhibit a normal morphology. Why does this happen? There's no symbiont to synthesize the contents of the blood meal to provide nutrition for the host. So the tsetse have no energy and no nutrients. So the youngs that they will try to produce will be deficient. This system is also been long established as vertical transmission. So with no symbiont to transmit to the larva, there's going to be no aid in larval maturation. So they will become sterile over time, as we do realize this system prides quality over quantity. This paper had recognized that Wigglesworthia is able to synthesize vitamin B6, which is an essential cofactor to its immune response. And also it synthesizes aminotransferase, which produces proline. Proline is an amino acid, and it is the main energy source for the tsetse fly. So without Wigglesworthia, there will be no aminotransferase synthesis of proline. Then there will be no soluble energy source for the tsetse. Just to reiterate my earlier point, why they produce sterile offspring, because if there's no energy source, no proline for the host fly, it cannot do these systematic processes to bring forth a successful young. So there'll be no energy source, no nutrients within the system, and a deficient immune response is not going to bring forth a successful offspring, and that's why we see them become sterile after generations. Now that we have covered the first symbiont in this system, we're going to move right on into the second one. So the second one is called Sodalis glossinitis. We're going to move forward and call it just Sodalis. This is a rod-shaped bacterium. It is non-modal and it is gram-negative. It is a microaerophile, so it relies on only a small amount of oxygen, roughly 0 to about no more than 10%. It is chemoorganotrophic and is the secondary intracellular symbiont in this system. And it does circulate in the hemolymph, which is the insect's circulatory fluid, and it's kind of similar to vertebrate blood. Getting into system discovery, there's no set established date. I did use as a reference point another system that Sodalis is in, and the Sodalis weevil system is. This system did date back to about 30,000 years ago. So in my system, it is older than that, but it is younger than the Wigglesworthia and tsetse system. So for Sodalis and tsetse, the relationship was described about 1993. They realized it was a secondary endosymbiont in 1998. The original understanding was that it was a commensal relationship with some association to trypanosome infection. They believed that Wigglesworthia played a role in trypanosome being able to stay prevalent within the fly. They did evolve their understanding to know that it is a nutritional relationship. It helps with immune system development and vector competence. Sodalis is a scavenger, as research has found out that it takes the rest of the metabolites that the Wigglesworthia synthesizes from the blood meal that the host does not pick up. It also does increase the susceptibility of trypanosome, and research has been trying to find ways to block the disease transmission. Now that we've understood that, we're going to talk about the transmission of Sodalis. So it is also seen as a vertical transmission route with the milk secretion similar to Wigglesworthia, but it was also seen to be a horizontal transmitted through mating. It is extracellular in the lumen of the milk glands, and also in the beginning of the presentation we said that Sodalis is also traveling around in the hemolymph of the fly, which is the lymphatics. It does secrete a recognition protein called outer membrane protein, or OMPA. This helps to resist the host antimicrobial peptides so that it can evade being degraded, and it also helps them to form a biofilm to colonize the gut. Now, how does Sodalis play a role in its system? So, Sodalis is going to provide the tsetse an attenuated immune response. It is going to exhibit quorum sensing, and it's going to provide acetylated homoserine lactone, or AHL. Quorum sensing is going to provide that chemical communication between the symbiont and its host, and the AHL is going to recruit symbiosis promoting factors and gene expression. When it does reach its threshold and there's enough signal, it is going to help the bacteria form a biofilm, and this will help it to suppress virulence factors to the fly and relieve host oxidative stress. So this is what Sodalis gives forth. This is its currency exchange in its system. And the TsetseFly, as we understand with most hosts, are going to provide a habitat and nutrients to the Sodalis. Now that we have a good understanding of the system, the host, and the two endosymbionts, we're going to take a look into vector competency. So we recognize that the TsetseFly is essential for the parasite's life cycle. In the cycle, it is the intermediate host. And in this system, it is specific as it requires the tsetse as a vector to transmit trypanosoma. The fly is refractory to infection. That means it's just stubborn to infection from trypanosome, and it has evolved its immune defenses to fight that parasite. Being a vector does mean that it is able to transmit disease without being infected. So getting into the life cycle, the infected vertebrate is bitten by the fly and is going to take up the trypanosome from that infected host. It is going to colonize the salivary glands and the proboscis of the tsetse, and then it's going to migrate to the midgut. So when it's in the midgut, it is now in its infected form and is called metacyclic trypomastigote. The trypanosome has parasite coats or surface molecules at varying levels of its development so that it can evade the proteases that are meant to degrade it within the host body. Variant surface glycoprotein, or VSG, allows for the parasite to be resistant to midgut proteases. It also allows for the parasite to undergo differentiation states. Studies have suggested that once the trypanosome colonizes the bacterium, there is a natural upper limit to how many can reside in this compartment, and this might be due to quorum sensing of the microbes, but they have not fully understood that sensation. Also, I do want to go back and say that now that the trypanosome is circulating within the tsetse's body, once it takes its next blood meal from an uninfected host, it is now going to exchange the trypanosome through the proboscis and get the blood meal in return, and then that's how it starts the cycle over. And then on the right is just a picture of different morphology of the trypanosome depending on where it is. So it does have different states depending on which is in the mammal, the salivary glands, in the TsetseFly, and in the midgut. So I just thought that was important to note that there's different stages of development, and then they have different infective levels. Now we're going to talk about Sodalis' specific role in vector competency. So the tsetse's midgut is layered with glycose aminoglycan, which is strengthened by the chitin, and this makes up for them lacking a mucus layer. This special layer allows for the midgut to be separated, with one compartment for the blood meal to go to, and another compartment that the trypanosome inhabits. So Sodalis secretes a protein called chitinase, which is going to degrade the chitin that the host puts up, and it's going to lead into a lectin inhibitory activity. So what does that mean? The chitinase is going to degrade the chitin, which is going to allow for the barrier to be weakened, and it's going to cause for the trypanosome to be able to penetrate through the midgut, and it can go from the one compartment that it's localized to, and it can go into the compartment that the blood meal is held into, and this is how we get that infection going on. The glucosamine is going to inhibit the lectin functions, which aids in killing the trypanosomes via apoptotic mechanisms. So when lectin is inhibited, it's going to increase trypanosome establishment as an inverse relationship. Flies with Sodalis are more likely to have trypanosomes because of this inhibitory activity that it displays. It is a parasite microspecies specific. We normally just see this with trypanosome and Sodalis. And there was a paper that talked about there's a threefold chance of infection when Sodalis is present. Now that we have seen what Wigglesworthia does for the tsetse and what Sodalis does for tsetse, I thought it was very interesting to see what both symbionts can do for each other. So this is going to talk about the co-interactions between Wigglesworthia and Sodalis. So papers have recognized that they have an inter-symbiont nutritional dependency. They are able to cooperate and they are interdependent on one another as they have co-evolved to live within this one system. And they have recognized that both are essential for the host survival and reproduction. So how do they have a nutritional dependency? Wigglesworthia, we know that it is going to synthesize the metabolites from the blood meal and produce B vitamins for the host. It also is going to produce thiamine, which is a vitamin B1, and this is the energy source for the Sodalis microbe. So the Sodalis is able to scavenge this vitamin from the metabolites that the Wigglesworthia synthesizes from the host diet. It also has a transporter mechanism to get these metabolites and be able to use it. And it also helps trypanosome spread because once it has the energy source, it is able to carry out its functions. There is a habitat partitioning, and this kind of helps to minimize competition. So Wigglesworthia is localized to the midgut, the bacterium in the midgut, and Sodalis is more so seen circulating in the hemolymph at any given time. And then they both, when needed, will be seen in the uterine pouch and the milk glands. So it minimizes competition. They don't have to compete for space because they both have their own partitioning habitat. And the paper talked about how the absence of Wigglesworthia will decline Sodalis prevalence over time because of that nutritional reliance. So we now understand how both of these symbionts play into the role, how the symbiosis has been established, and how they work well. But what happens when the system is disrupted? So we're going to take a look at both sides, some imbalances, and how we can bring back an equilibrium. So starting off with Wigglesworthia, remember we talked about this recognition protein, PGRPLB. When it is gone, the absence of it can harm Wigglesworthia because it helps to be recognized as a friend by the host to resist the antimicrobial peptides. Without PGRPLB, it's not going to have anything to say, hey, I'm a friend here, I can help you, and it's going to get degraded. It also has a multivitamin transporter, SMVT, that helps to transport the vitamins across the cell membrane. So if the transport is not present, the bacteria cannot do that. And we're going to see a disruption of metabolic pathways, specifically with carbohydrates and amino acids being deficient. We're going to see a loss of fertility. We're going to see that it cannot feed the larva because there is no, the food is not being broken down so that the host can intake it. So if they can't feed the larva, they can't feed themselves, we're going to just see deficient host nutrition. We're going to see a decrease in successful offspring. And we also talked about proline not being produced, which is the energy source for the TsetseFly that Wigglesworthia also synthesizes. Another disruption can come from antibiotic treatment, specifically with tetracycline, doxycycline, things of that nature. It really does disrupt and damage Wigglesworthia, and the absence of that will cause severe ramifications in this system. How can we bring back some maintenance? The paper did talk about allowing for more vertical transmission to happen to kind of replenish the Wigglesworthia population. It can also supplement some B vitamins to the host. So the Wigglesworthia can just recuperate and try to regenerate. So it's not going to disrupt the whole system, but they will allow for that microbe to just rework itself back into the system. Also, the last thing was to also block the host immune response so that it does not further degrade the amount that is left. So it's just some preservation methods. Looking to the other side, Sodalis, it recognizes that thermal stressors above 30 degrees Celsius can disrupt Sodalis. In the gut microbiome, the changes also can disrupt it. And this part I thought was very interesting, how the blood meal raises the host temperature, and thermal stressors is a problem to Sodalis. We see when Sodalis is disrupted out of the system that there's a decrease in trypanosome circulation because there is an increase in lectins in the body. And overall, without Sodalis, we're going to see a decrease in vector competency. So how can we bring back some maintenance to this system? Temperature control, and one paper did an experiment how they just inoculated Sodalis back into the TsetseFly, but that would not really work in a naturally acquiring system. That is more like a manipulated reintroduction. So the first topic that I decided to research that was of interest to me was how the immune response plays a role in this system. So first we're going to talk about just the general immune response that the tsetse displays. So it has this protein called opsonins, and opsonins are going to coat the pathogen and mark them for engulfment by the hemocyte. And a specific opsonin that this system has is the thioester containing proteins, or TEPs. This is to prevent systemic infections. So the fly is going to release opsonins to prevent systemic infections. It is a part of its innate immunity, and the TEPs are going to bind to pathogens and act as a ligand, and then that ligand is going to bind to the phagocytite receptors to activate that process. This is going to ensure efficient pathogen removal and protection of the tsetse fly. This is also crucial as the fly is a vector. So if the fly is infected by another pathogen, then it cannot effectively carry out its role as trypanosome's vector. It does display tissue-specific expression patterns, and we see that opsonins are upregulated with trypanosome infections. It does regulate the immune response to favor the symbionts. I thought this was cool because of that recognition protein. It plays such a big role in this system. I would think it's an essential component because it's going to cause the fly to change the response that it gives, and it's going to prevent the aggressive antimicrobial response that would decimate the Wigglesworthia. Now we're going to take a closer look at Wigglesworthia's specific role within the tsetse's immune system. So it's going to promote development and function of the system. When it is gone, we see that it compromises the immune system. There was a study that performed an experiment to look at the effects of the tsetse fly with the absence of Wigglesworthia. So they studied wild type and an aposymbiotic tsetse, so one that used to have all of its symbionts and then has been cleared from tetracycline, which is an antibiotic treatment. They also treated it with non-pathogenic E. coli, and they were able to realize that the flies that are aposymbiotic were quick to succumb to infection. They saw that in those aposymbiotic tsetses, they had absent phagocytic hemocytes and atypical expression of immunity genes. When they wanted to reverse the phenotype, they were able to transplant hemocytes back into the system. And they gave it yeast as well as tetracycline because they said the yeast was to help to prevent the fly from being sterile. They just wanted to see the fly without its symbionts, but they did not want to disrupt the system altogether. And going to the right side of our things, on the two figures, the first one is showing the circulating hemocytes within the system and sessile, which means not moving, like not readily available hemocytes. And they looked at the wild type and also the aposymbiotic tsetse, and they see a very big decrease in hemocytes in the circulating and the sessile when the fly was without its symbionts. So this kind of helps us to know that the symbionts aids in hemocyte prevalence and maturation, which we will get a little more into in our further slides. And then at the bottom, they did a line graph comparing the percent survival rates, looking at 3 different graphs showing the wild type, a fly without its Wigglesworthia, and then a fly without just an aposymbiotic fly. And they injected the E. coli, and they also did per os, which means orally given to the flies. And we see that there is a decrease in the survival in all three graphs. But if we take special look at the aposymbiotic graph, the fly that had the 10 to the third CFU of E. coli injected ended up quickly declining and eventually died by day 12. So the paper didn't really touch too much on it, on the injected versus the oral. But the main takeaway from this graph was just to show that there is a decrease in survival when the fly is rid of all of its symbionts. It is unable to produce a stable and effective immune response. So we talked about this in the past few slides, but I do want us to get a better understanding of it. So what are hemocytes? They are immune cells in the vertebrates, the white blood cells. They're going to circulate in the hemolymph or the lymphatic system. They aid in defense and development of the fly. They have some qualities to it. They can do phagocytosis, they can do encapsulation, and they can do melanization. And melanization is the process of creating melanin to kill pathogens and to seal wounds. Hemocytes, the production of them and the success are heavily dependent on Wigglesworthia, as Wigglesworthia does aid in developing the hemocytes during larval stages. The blood meals are going to activate hemocyte differentiation, and without it, the flies are more susceptible to infections. The next topic that I was of interest to me and that I studied was Wolbachia. So Wolbachia pipentis is the secret third symbiont within this system. This whole time we did touch on two, but the fly is able to live in harmony with all three. So just a little overview. It was first reported in 1993. There are many strains of tsetse-fly, but we only see Wolbachia in Glossinia mortisans, which is the strain that I have been studying, and also mortisans centralis species. A paper did talk about how geography influences Wolbachia's presence, as there is about 10 different African countries that has a tsetse-fly, and we also see Wolbachia have different rates of pathogenicity and infectiousness in these different strains. So in one country it can be closer to 100%, and one it will be maybe 80 or 60. So it just has different pathogenicity rates in different geographical areas. Wolbachia is the reproductive symbiont in this system. It is an obligate intracellular, and we see that it characterizes in the germline, specifically the oocytes, spermatocytes, and we also see it again in the uterus pouch and the milk glands. It is maternally transmitted, or as we have came to understand, vertically transmitted through this germline. And how does it play a role in reproduction? It's going to induce pathogenesis and feminization, which is the killing of the male insects and making them sterile. So Wolbachia is inherently pathogenic by nature, but there are certain strains that is mutualistic to its system. And this new phenomenon, or relatively new phenomenon that is going around, is using Wolbachia as disease resistance measures. And how can they do that? So some researchers have genetically modified Wolbachia, and it can interfere with pathogen replication. It can also decrease trypanosome development by expressing anti-parasitic genes and outcompete other viruses for resources. So these are just a few ways how it can be used as disease resistance measures. So what are some techniques that researchers have identified to successfully reduce Wolbachia's prevalence? The first one, which is a highly effective method, it is tried and true, it's a little bit older, it's called the sterile insect technique, or SIT. So the males are going to be chemically sterilized or induced with UV radiation, and this is going to help suppress the female fertility, because if the males are sterilized and they end up mating, they are not going to produce viable young. Or if they do produce young, is not going to have Wolbachia expressing the pathogenic disease traits. So this was the first method, but they did come up with a newer method, which is called cytoplasmic incompatibility, which is going to lead to embryonic death. It is a reproductive manipulation tactic that is produced by the Wolbachia, and when they do certain crosses with the strains, it is going to lead to that death. The females are going to inherit the mitochondrial DNA and the Wolbachia bacteria, and that's why this doesn't really affect the females. And it's going to push and drive desirable traits and release that out into the naturally occurring systems. So just taking a quick look onto the right side, it is a Punnett square showing two different ways we can do CI, which is the incompatibility. So unidirectional is when an infected male crosses with an uninfected female. And we're see that when the male is infected, the female's uninfected, that's when we are going to not have a successful offspring. But even if the female is infected, they can still produce a viable offspring, which I thought was very interesting. That the female, it does not regard her status, whether the offspring, whether it will be viable or not. Going to the other side, which is the bidirectional, is when the different species cross are incompatible in both directions. So going back to our chart, the white is the uninfected, and the blue, the light blue, and the black are infected, but with different strains of Wolbachia. So it does, like we said, it does not matter if the female is infected. However, if her strain is different from the male strain, it is going to cause that's the offspring to not be viable. So going through the chart, we have infected Wolbachia female and also infected male, but it still produced a offspring, and I just think that that Wolbachia strain, that black one, is still compatible, and that's why they were able to still produce a successful offspring. However, when it had the uncompatible cross, which is that light blue, dark blue, we were unable to get a successful match. But when we see the light blue female paired with the light blue male, again, we're going to see success. Just kind of going back to the unidirectional, light strain is going to produce success. So this is just a newer method that they came out with that they seem that is works within the systems. This is just going to help to reduce the vector transmission overall. So they came up with these methods because we cannot cure Wolbachia with antibiotics simply because we risk destroying Wigglesworthia in this system. So they had to resort to these methods to reduce pathogenicity without creating aposymbiotic tsetses. And the last thing I just want to reiterate and or clarify is that the two infected hosts can still produce viable offspring because the female's bacteria and her mitochondrial DNA can rescue the modifications going on by counteracting the infection. Just to wrap it all up, we're going to talk about what we've learned throughout this presentation. So the TsetseFly is the host in this system, and it is the intermediate host of African sleeping sickness, or T. brucei. It does have special mechanisms so that it does not get sick or infected by trypanosome. It has a midgut with two distinct layers, and that's how it is able to keep the infection at bay. So the first endosymbiont in this system is Wigglesworthia, which is we recognize to be a crucial component in this system as it aids in immune defense. It allows the fly to regulate its immune response when it is recognized with its recognition protein. It does help in nutrition acquisition as it's going to synthesize metabolites for the host and for the other symbiont in this system, Sodalis. And it helps in embryonic development as it is passed on vertically transmitted to the young, and it helps to successfully grow the larva in that stage of life. The second symbiont, Sodalis, is also crucial in this system as it helps to aid in trypanosome spread. We talked about chitinase and how it degrades the chitin, which is there as a protective barrier to the midgut. And when the midgut is penetrated and the trypanosome is able to colonize, this is what causes the problem. And we are able to see trypanosome circulating in the fly. And when it goes to take its next blood meal from an uninfected host, if the fly is infected with trypanosome, it is going to pass it on to its next victim. Some other things that Sodalis does is it plays a role in the immune system. It is also going to be evaded by the harsh antimicrobial response because of its own recognition protein, OMPA, and it allows for the immune system to be altered to favor the symbionts so that they are not degraded when the host recognizes a foe or a pathogen in its body. So we understand that the currency exchange between the symbionts to the host, but there is also a currency exchange or a co-interaction between both symbionts, Wigglesworthia and Sodalis, and they do display interdependency. Mainly Sodalis is dependent on Wigglesworthia for scavenging of thiamine, which is vitamin B1, which is the energy source for Sodalis bacterium. So there can be instances where this system does fall apart through dysbiosis. Talking about Wigglesworthia, antibiotics can be a factor. Its recognition protein being gone can be a factor, as there is no distinction between friend or foe. So the host is just going to degrade everything as it regards it as a pathogen. With it gone, also there will be loss of fertility, and we can see that the adult fly will essentially become sterile. On the other hand, Sodalis can have dysbiosis with thermal stress and a decrease in trypanosome circulation. And there we talked about ways to bring that maintenance to maintain equilibrium within the system. We touched on how Wolbachia is a third symbiont within this system, and it is a pathogen by nature, but can be mutated to be beneficial. We talked about the sterile insect treatment or technique that was an older tried and true method that was allowed for sterilization of the male insects. But then there is a more newer advanced thing that they're coming up with, the cytoplasmic incompatibility, and irrespective of the female's position, they can still produce a viable offspring. So we see that the male is what is the driving force between successful or unsuccessful. So they are trying to create these different strains and inject it into the male tsetses, which they call feminization, and they can push desirable effects into the population. And as a result, try to use that to decrease pathogenic spread, but in our system specifically to decrease trypanosome spread. So this is how they want to decrease African sleeping sickness spreading within those endemic areas. And then antibiotics is advised against because it would rid this host of its needed symbionts. And we kind of talked about that in the dysbiosis, and also in the immune response, how when it is absent, it causes a whole lot of ramifications within the host fly. So my one sentence takeaway is that the TsetseFly needs all three symbionts for effective functioning. Thank you for listening to my presentation. I have learned a lot over the semester, and yeah, this was a great project.